Hi everyone, my name is Maria, and today I'd like to tell you a little bit about my proposal for Yukon PostDoc Seat Award. Unfortunately, we're still in the middle of the COVID-19 pandemic right now, and one of the things that's been highlighted during this pandemic are differences in individuals who contract the virus. So, for example, we've probably all known some people who've tested positive for the virus, and they're asymptomatic or have very mild symptoms. And then unfortunately, there are other people who contract the virus, test positive, and they end up in the hospital. They end up with what's being termed long term COVID, or they even succumb to the disease. And on the vaccine side of things, we're also seeing differences in how individuals react. So, one example is that my mom and I both got the Moderna vaccine. So, she had no side effects after the first or second vaccine. She was outside gardening and cutting the grass and just doing whatever she wanted. I, on the other hand, did have some side effects after the second vaccine. I had arm pain, I had fever, I had chills, and other flu like symptoms. So, why is this? Well, the immune system has natural variation built in so that individuals and populations might react slightly differently to the same threat. And this is so it protects species from going extinct due to a virus, a bacteria, a parasite, or something else that might be harmful. We also see this natural variation in humans too, even in fish, which is what I study. So, my research organism is a small fish called the three-spine stickleback. And yes, you can find it right here in Connecticut. What makes this particular fish so interesting and important to study is that different populations of this fish react differently to a naturally occurring parasitic worm. So, you have some populations of this fish species. They're exposed to this parasitic worm, it gets inside of their body and it grows and grows and grows. And when you dissect the fish, as soon as you open up the body, the worm is right there. These worms can grow so large that they can be up to 1/3 of the body mass of these fish. So think about your body mass, think about 1/3 of that, and having a worm inside of you. It's wild, right? But on the other hand, some populations of three-spined stickleback are able to suppress the growth of this worm. So when you open up and dissect these fish, you can barely detect that worm with your naked eye. So how does this work? The truth is that it's complicated, and we're still trying to tease apart all of the factors that go into these differences. But we do know that one important component of these fundamental differences is how immune cells look and react in different populations of these fish. This is also true for humans and other animals too. So, with this grant, I want to dig down into this immune cell variability. This will involve isolating at least two different types of immune cells from at least two different populations of three-spined stickleback. And once I isolate these cells, I want to learn exactly what conditions make them happy and reproductive, and then test these cells to understand the differences in how specific immune cells isolated from different populations react to a stressor. Do they react similarly? What's different? And really dive down into that line of work. So, just like how you and I might react to a stressor differently, fish and other animals are the same way too. And cells can really tell us a lot about how we react to stress. So, not necessarily, the stress of giving a presentation for a potential award, but the stress of virus, a bacteria, a parasite, or other stressors like that. So, if we know more about how these cells impact the way we react to stress, or these fish react to stress, we can learn a lot about the evolution of these cellular differences that have occurred and what the consequences of these differences might be. And the importance of this research is that the more we can understand about the link between individual cellular variation and disease, the more that we can work on treatments, therapies, and preventions for animals and also for humans. And on a personal note, this research is important to me because this presents an opportunity for me to build a living library of various types of immune cells from these three-spine stickleback populations. It allows me to generate my own protocols that I can share open access with researchers around the world. And all of this can be leveraged into larger grants to build my career as an independent researcher. Thank you so much for taking the time to listen to me today, and please stay well.